of elk. So elk are the primary thing that wolves eat. They're basically big deer, and their populations were pretty dense and large, and they were eating a lot of the plants, the sort of foundation of the ecosystem diagram that I showed you. When wolves were reintroduced, the story goes that they caused changes to either the numbers or the behaviors of their ungulate prey. An ungulate is an elk or a deer or a moose, pretty much a hoofed animal in the deer family. The reintroduction of wolves caused changes in the numbers or behaviors of their prey, which then resulted in these animals not just sort of hanging out, eating a whole bunch of vegetation, which then resulted in regrowth of vegetation like willows along stream banks and aspens and cottonwoods. And so this is a very nice, tidy story. And because of this, we now have this sort of idea that bringing a top predator back into the ecosystem will restore healthy ecosystem function. However, this story is missing a key player, and you can see that player in this picture on the left. It's people. And while we've learned from this reintroduction that the behavior of wolves can affect the behavior of their prey, which can affect other things about the ecosystem, we also know that people affect the behavior of wolves and their prey. So now that wolf and elk and deer populations have expanded well outside the sort of small special protected area of Yellowstone National Park, they're increasingly coming into contact with a rapidly growing human population. So these wolves are out on the landscape across huge areas where people live and work and recreate now. So what we don't know is whether and how we expect these predator-prey relationships and their associated ecosystem functions to play out in areas that are more human-dominated. So this is one of the things that I'm trying to understand with my PhD research. And to sort of start wondering about this, I asked the question, how might humans affect the behavior of wolves? Well, we typically think that wolves, want to avoid humans. And so perhaps they do that because humans put out physical infrastructure on the landscape that's unnatural, things like paved roads. So perhaps wolves are avoiding just this unnatural physical infrastructure. And if that's the case, we would expect wolf use of the landscape to be primarily influenced by the locations of things like buildings and paved roads. Or maybe it's not physical infrastructure, it's something about humans themselves, perhaps some kind of noise or activity disturbance that might cause wolves to avoid human areas. And if that's the case, we would expect them to respond not only to buildings that humans are around, but also to travel corridors like ones shown here. And we might also expect them to act differently during different times of day, because we know from other studies that animals will often become more nocturnal to avoid coming into contact with humans while they're active on the landscape. Or maybe wolves aren't just disturbed by humans, maybe they actually perceive some type of risk from humans, a risk of mortality. If that's the case, we would expect the way wolves use the landscape to be influenced by whether or not humans hunt them. And then there's this sort of alternative idea that a lot of times we see these big concentrations of deer and elk down around human areas, especially when humans provide them food. So if humans are concentrating prey in these sort of predictable areas, then perhaps that would actually cause wolves and other predators to be drawn to those areas. So if that's the case, we would expect places where food is provided to ungulates to influence where wolves are located on the landscape. And then, of course, there's the possibility that humans don't affect the behavior of wolves. They don't care at all, in which case we would expect their use of the landscape to just be primarily determined by things about the natural environment. So each one of these kind of pictures here represents a hypothesis that we would like to test. And the way that we can test these hypotheses is by trying to tease apart the influence of each one of these predicted things on wolf use of the landscape. I'm seeing chat things and I don't know if I'm supposed to be, recording, excellent. Okay. So I am studying this question in Jackson Hole, Wyoming, which is sort of the perfect place to tease apart these different hypotheses. So Jackson Hole is just south of Yellowstone. Wolves naturally recolonized the area after being reintroduced up into northern Yellowstone here. And Jackson, the town of Jackson is sort of this concentrated, urbanish type of area. I'm sure none of you find it urban, but for the West, for the Intermountain West, it's fairly urban. So we have this sort of denser town area, and then these are the Tetons here. And as you move north up the valley, you can kind of see this like suburban and exurban type developments surrounded by big swaths of undeveloped land. So here we have every type of human influence that we'd like to tease apart to understand what it is about humans that might influence the behavior of wolves. We have that physical landscape conversion. We have also a huge recreation and tourism industry, so lots of human activity out on the landscape. There's been intermittent wolf hunting, and the National Elk Refuge is located in Jackson Hole. So we know where food is actually being provided to elk and other prey. So this is sort of a bird's eye view of Jackson. Again, these are the Tetons here, and this whole basin is kind of referred to as Jackson Hole. And so the data that we can use to test these hypotheses are, we have wolf GPS collar locations. So I'm really lucky to work closely with researchers at Grand Teton National Park and Wyoming Game and Fish, and they have shared their collar location data with us dating back to 2005. And so we have locations for 58 individual wolves in 15 packs across this landscape. We also know where human influences occur. So we know where buildings are located. We know where elk and other prey are being fed. We know where roads are and where travel activity corridors are located. And we know the years during which wolves are hunted. So these are all of those things that we predicted might influence wolf use of the landscape. We also know things like how deep the snow is and how rugged the terrain is. So we can also incorporate that into our understanding of how wolves move around. So really briefly, kind of what we do is we get these data and we develop statistical models that help us relate wolf locations that depict how wolves are using the landscape to different environmental and human related factors. And we see how well those environmental and human related factors kind of explain the variation in wolf locations that we see. And we can come up with different models that correspond to each one of the 5 hypotheses that we're interested in testing. And then we compete those models with something called AICC model selection. And by doing that, we can see which hypotheses are better supported by the data that we have. And I'm going to yada through a lot of things and tell you that after we do all of this, what we've learned so far, we found the strongest support for the activity, disturbance, and threat of mortality hypotheses. Specifically, we found that wolf use of the landscape is influenced by the location of buildings and travel corridors, not just paved roads, but corridors that humans are active on, and that they change during the time of day, depending on whether it's day or night, and whether they're being hunted. And in a very small nutshell, what we found is that wolves appear to be able to alter their behavior to still use human areas, specifically by moving closer to human areas when humans are least active and when humans are least directly threatening to them. And I'm not going to go through graphs for the sake of time, but I'm happy to take questions if anyone's really interested. The take home here is that, we're wondering how these sort of predator-prey relationships are playing out in human areas. And what we're finding from this historic wolf collar data is that wolves seem to be able to alter their behavior to still use human areas while avoiding coming into contact with humans. So then the next question that sort of inevitably builds from this is, okay, well, if wolves can still use that area, does that mean they're still affecting their prey in the same way? And to understand that, we need to know whether humans change not just where wolves are on the landscape, but how effectively they're able to kill animals on the landscape. And we can't understand how humans change predatory behavior of wolves just by looking at GPS collars. So this is the field work that I'm currently working on, is learning more about what wolves are killing and where they're killing it. And you can do this in 3 easy steps. Number 1, you put collars on wolves. Step 2, you use those collars to figure out where wolves are spending their time based on where you have kind of locations clustering together. And step 3, you go into those areas and you use context clues and the landscape to determine what has happened in those areas, what kinds of activities the wolves were doing. And if we find a predation event, which is what we call, when we find something that wolves have killed, we get information about that. So we find out what species it is, what sex it is. We take samples to determine how old it is and what body condition it was in when it died, and we also take down information about the environment. And so we've been doing this for the last 2 winters, and this January coming up will be our 3rd winter. So far, we've investigated more than 600 wolf activity areas. More than half of them were places where wolves sleep, but we found a little over 100 kills. And so we anticipate after this winter, we should have enough information to run these models. So because the models are a little bit complicated with the number of things we want to look at that might influence wolf predation behavior, we need to have a lot of data to put into them just so they kind of work statistically. And so that's why we're doing this for 3 full years so that we can have decent confidence in what we find out from our models. And if we can build these more complex models, then we can compare, how wolf use of the landscape differs to how wolf actual predation events differ around human-dominated areas. And when we find out whether humans are actually reducing the risk of predation for ungulates, for wolf prey, then we can continue to build on that. So if we find out that predation risk is lower in human areas, then we can build up to ask, what is it about humans that causes this? We can ask whether certain types of animals benefit more than others. So we can start thinking about how relationships in the food web might change if, say, deer or elk or moose are benefiting more from these human-provided refuges. Alternatively, if humans aren't changing the risk of predation for ungulates, then we can investigate, we can drill down deeper into wolf behavior to see how they're altering it to still kill prey around humans. And also, we are now fortunate to have wolf and elk collar data in the same place and time. So we can also look at interactions and kind of simultaneously and see how prey like elk actually act to perhaps reduce their own predation risk around human-dominated areas. So the overarching goal here is to get a more complete picture of these predator-prey relationships actually in human areas in the real world where these species are now living and flourishing, with the overall goal of sort of filling in this question mark here and helping us understand how we expect these important predator-prey interactions and their associated ecosystem functions to actually be playing out in these human-dominated areas. So that is my research in a nutshell. I'm happy to take questions now, I think is how we're doing it. And you're also welcome to email me if you think of a question later.
Great, Kristin, thank you so much. This was super cool. Questions are happy to come through the chat or the Q&A section or also through Facebook. But if I can start one off. So if you're looking at kind of how humans might affect, like, wolves are only coming into human areas at night. Are you seeing at all their prey? You'd think like their prey would then kind of be safe from wolves near human areas in the daytime, but obviously a threat, of human human development to their prey as well in the daytime. Do you see kind of like elk more frequently in human areas during the daytime versus nighttime switch from wolves, or does kind of the fact that like cars and buildings and other stuff are around dominate?
It kind of depends. And their behavior's been changing in recent decades. So over the past few decades, we've seen more elk and deer kind of hanging out around, in town, basically. But also they seem to have this they typically have a kind of cycle where if they're going to be very close to humans, they usually would come in and do that at night. So they would like move in and eat at night and then go back into the hills when humans are more active. And so, yeah, it'll be interesting to see how those different daily activity patterns of wolves and elk sort of both simultaneously change in these human areas.
Cool. Jenna, are you seeing anything from the Facebook side? Okay.
No, but I also have a question for Kristin. So this kind of reminds me, I guess, of like with the bears in Yosemite, I guess, like, and since they're also super predator, that their behavior has definitely changed. Like there's always warning signs about like, don't leave your food. And I was just curious, like I know wolves and bears are very different animals, but do you think that there would ever be the possibility of some sort of study to like basically compare how these two predators have had their behavior affected and changed by humans, and to see if there are any like trends or relationships?
There are a couple of people trying to look at that. It gets really complicated trying to look at multiple species in the same study because the way that you capture animals and color them, yeah, it's harder to color a grizzly bear, I would say. But people are kind of looking at it. I have a friend of mine is looking at how both of those animals are changing patterns of mountain lion behavior. They're kind of like the step below. But generally, I mean, grizzly bears and wolves don't so much get in fights with each other, or like they don't seem to change each other's distributions that much. In fact, in the spring, we have to be really careful because grizzly bears are always trying to sit on the carcasses that the wolves have killed. So they're often found in the same areas, kind of eating the same things.
Okay, thanks.
It looks like we also have a question in the chat that says, have there been any unexpected consequences of reintroducing wolves in the Yellowstone Jackson area, or unintended consequences?
I mean, there are certainly a lot of consequences for the
All right.
I know we might have lost Kristin for a second.
All right. Olga, we'll get back to that question hopefully when Kristin has better internet connection. And also, of course, any further questions for Kristin can be left after the chat. I think our main problem is that screen is being shared. Kelsey, do you know if there's a way to
I think if Kiera wants to try, okay.
We can override. Okay, then we'll have, hopefully Kristin will come back to us at some point. We can finish up questions. And also, if she does end up being around, she will then, of course, be around a little bit at the end for questions, of course, if you have those later. So for now, Kiera, if you can share your slide. So our next speaker is Kiera Wilhelm, sorry. She's coming from the UC Berkeley chemistry department, where she studies the immune system and T cells. And the thing that she finds most interesting about this kind of is that we really have no idea how T cells are activated and how they work in your immune system, kind of to, fight pathogens or other disease-carrying molecules. But they're super, super critical to how our immune cell functions. So she's here, of course, to talk to us today about kind of, what little do we know, what is there to figure out, and why are T cells so cool? So we're happy to hear about it. And if you have any questions, feel free to leave them and we'll get to them hopefully in short time. All right, Kiera, take it away.
Awesome. Thanks so much for that introduction. So yeah, my name is Kiera Wilhelm. I'm a fifth-year graduate student at UC Berkeley in Jay Grove's lab in the chemistry department. And the title of my talk today is How We Use T Cells to Protect Our Health, Even If We Don't Quite Understand Them. And because it is relevant to all of us, I'm going to introduce T cells and how they work in our immune system in our body. First, by talking about these new mRNA COVID vaccines, and just to use that as a springboard for talking about the immune system. And I'll also discuss some developments in using T cells to fight cancer and developing cancer immunotherapies. But really what I study is what we don't understand about T cells, what we don't understand yet. And so, yeah, then I'll get into more what my research is and how this fits into the developments in health and medicine that are already taking place. So the mRNA vaccine for COVID, there's been a couple that have recently come out with very surprisingly good efficacy. And so I'll want to explain to you all how these vaccines are working and how they can trigger our T cells in our immune system to fight a potential COVID infection. And so these vaccines, let's see, do I have, yes, I have a pointer, are the messenger RNA is instructions that genetic instructions that can be inserted into our muscle cells, be taken up by our muscle cells and translated by our cellular machinery to make the spike protein that we see on the structure of the coronavirus. And so we're using our own cells to manufacture this protein portion of the coronavirus. And then these cells will excrete that into, say, our blood system. I am getting the spinning wheel of death, which means I am not able to advance my slides. Let me try to stop sharing. Okay, that's fine. I might have to reboot PowerPoint. Sorry about that. And then I find Zoom. Okay. Okay, we're back. Yeah, and so then we have these spike proteins that are floating around. And so what happens with these? And that's where our immune system comes in. So these viral proteins can get picked up by these immune cells that in general, we'll call them antigen presenting cells. And they're cells that take up bits of protein in either just directly or in the case of a B cell by using a specific receptor. And then they chew up the proteins that they find. And then these little bits of chewed up proteins we call antigen, and they will be presented on the cells that picked them up. And so say we have this antigen presenting cell that is presenting this protein fragment or peptide in an MHC molecule. And so this is the signature that will be recognized by the T cell and its T cell receptor. And so then the T cell will say, oh my goodness, wow, we have this protein that's been floating around, this peptide fragment. That's bad. And so it gets signals to replicate, make more clones of itself so that lots and lots of T cells can go out and try to fight this viral infection, or be made so that in the case of a future viral infection, they'll be ready to go. And then these B cells, similarly, will present the peptide in an MHC complex. T cells, in this case, it's a T helper cell, but that's more detailed than we need, will recognize this peptide in the MHC complex. And this will actually, this interaction can signal back to the B cell and say, oh my goodness, you found something bad. Whatever antibody, whatever receptor you used, make more of that. And so then this B cell transforms and starts cranking out lots and lots of antibodies that are able to recognize the protein. And so this is really a part of the pathway that is how our immune system will recognize these proteins that are given to us through the vaccine and mount an immune response either toward an infection or in preparation for a future infection. And so then the T cells can go out and actually be ready to kill infected cells. So they are ready with this T cell receptor that recognizes, in the case of COVID, the spike protein and some protein fragment. It says, oh, I recognize you, you're something bad. And then the killer T cell will kill that infected cell and clear the infection. And then the B cells cranking out antibodies then can neutralize the virus. And hopefully, through this process, we become healthier through time and the virus gets cleared. And so I want to show a video of this T cell interacting with an antigen presenting cell. This is taken using a really nice microscopy technique that allows good resolution in 3 dimensions with good ability to see in time. And we can see this T cell landing on the antigen presenting cell. And you can see it's quite an active interaction where the T cell is probing the surface for those peptides. And then in this case, this T cell saw an antigen and it says, wow, I need to get more information about that. And so it has a prolonged interaction in which the T cell gets activated from the peptides presented on the antigen presenting cell. And then T cells, not only can they clear viral infections, they can also kill cancerous cells. So, and this is not really just, not necessarily a big dramatic case of cancer. This could be a cell that somehow is misfired, misfunctioning, and it's very normal for T cells to just identify these peptides, say, that's not normal, and then kill the abnormal cells so that it doesn't turn into a greater tumor and become a more dramatic health problem. And so we can, the T cells are used not only to recognize foreign things coming into the body, but also help clear our own cells when they start acting out of line. But then what, we can still use these tools at the T cell tools, or we'd like to use the T cell tools to deal with more persistent tumors or cancers that are resistant to, say, radiation, chemotherapy, other therapies. And so a lot of recent efforts have gone toward directing T cells to fight cancer. And so, what if we don't have any T cells that recognize a specific tumor marker and we have this persistent tumor? Is there some way that we can change what's happening in these T cells so that we can direct them toward that tumor marker and have them clear the cell and specifically target these tumor cancerous cells and leave our healthy tissue alone? And there are a couple ways that researchers are figuring out how to do this. One is to introduce an engineered receptor into the T cell. And this engineered receptor is designed so that it can bind this specific tumor marker and hopefully transmit similar signals to the T cell that will tell the T cell, this is bad, kill this tumor cell. This is quite an invasive process. It requires taking a whole bunch of T cells from a patient, then genetically introducing a new receptor into the T cell, growing those up, and then reintroducing this new T cell population back into the patient. And these therapies exist and have been used to some success. Another alternative way to direct T cells to fight these cancerous cells is to use some sort of linker molecule that links the tumor marker to the native T cell receptor. And surprisingly, it might seem strange that just linking the receptor, not necessarily docking correctly on the receptor, is enough to activate the T cell and say, hey, kill this tumor cell. But these linker molecules actually are quite effective. And it really leads to a lot of questions about, well, if you can do all these strange things to activate the T cell, then how exactly is the T cell being activated? How is this T cell receptor being triggered? And the thing that I find the most wild about this field is that these technologies are being developed and used to help patients. But the basics of how a T cell receptor really gets triggered by its antigen is really not known. We don't know. And so those are the questions that we are asking in our lab. So first, how do T cells know the bad stuff from the good? So far, all of the peptide in the MHC complexes that I've been showing have been bad. In the T cell, you want the T cell to respond. But the peptides that get mounted in these MHC molecules are not necessarily bad or foreign or cancerous. Many normal protein fragments get mounted into these MHC molecules. And the self molecules are actually about on the order of 10,000 times more abundant. So let's see, the non-self protein, the non-self, this foreign peptide is 10,000 times less abundant than the self, and only about 10 times more potent. And so that means that this T cell receptor can interact pretty well with all of these self peptides. It just interacts better with the foreign peptide. And so this is sort of like looking for a needle in a haystack. And there's just so much hay that even though we know that a needle looks different than the hay, it takes a lot of searching and a lot of time to find that needle. And so we want to know, how does this T cell process all of these inputs, and what signals does it require in order for the T cell receptor to say, yes, that one is bad, T cell, I want you to activate. And so my research really focuses on how does a T cell receptor get triggered. And there's a hypothesis that T cells might use a stopwatch in order to decide what it should respond to and what it should not respond to. And so here's a little animation that we have here, a self peptide. It comes down, finds the receptor for a certain amount of time, and then leaves. But it hasn't reached the amount of time necessary to stick around in the receptor for the cell to actually activate. Another receptor might come along, bind also for a short amount of time, then leave. The cell still hasn't triggered. And then another peptide, PMHC molecule, might come around and bind for a long enough time that cell signaling happens. The remarkable thing that I really want to point out is that here we're talking about a single receptor measuring how long a single molecule of agonist or of this foreign peptide sticks around. There are tens of thousands of molecules on the surface of this T cell, and somehow it's able to coordinate and pick out information from one particular receptor that bound a ligand PMHC molecule for a long enough time. And so what is this stopwatch, and how is it possible that this stopwatch is so sensitive to these single binding events? And so we answer that question or address that question using a microscopy setup. So in our experiments, we make a synthetic antigen presenting cell where we put our PMHC molecules. And then to this bilayer, we introduce T cells that have a T cell receptor that we know binds to this PMHC. And then we track the localization of various signaling proteins within the cell. So in the movie that I'll show, you'll see single red spots appear. And these are single molecule interactions between a receptor and a PMHC molecule. And then you'll see a background green protein. It's imaged green or colored green in this video. That is diffused throughout the cell membrane, but then will cluster on top of locations where the peptide MHC has bound the T cell receptor. So here, that's the cell landing on our bilayer, and you see it's picking up these single molecule binding events. The signaling cluster, the signaling hub molecule is clustering. And then the cell continues to spread around. It gathers all of its binding events in the center. We'll let that play again. So you can see these green clusters firing if the binding events stick around long enough. And so our lab is hypothesizing that it's these signaling clusters that are serving as the stopwatch for the cell, and that the single PMHC molecules must bind for a long enough time in order for that cluster to form, and that will allow signaling to happen further downstream for the cell to say, hey, I found something bad. Now I should do something about it, like kill this cell or activate this B cell to have it produce antibodies.
Hey, Kiera.
Yeah.
I just have a question. Hopefully it's like a broad one about like, what exactly is like, I guess, the cell signaling event? Is that like, I guess when it's activated, does that mean there's just some sort of like electrical signal that's sent through to other parts of the cell, or?
Yeah, so what we're seeing with these, let me go to a frame where we see lots of them nice and bright. So this is what we're imaging is a protein that's very abundant in the cell. And we see it, get very bright. A lot of this protein enters a very small area. And we're imaging a particular scaffolding protein, but there's a whole network of signaling stuff that's within these clusters. Physically, what might be happening is a phase transition within the membrane of the cell, that proteins are actually entering a different phase in which they're more gel-like instead of liquid-like. So that could be the switch that's controlling whether the signal goes through or not. But that's another thing that our lab is investigating, is really what are the properties of this cluster and how could they be controlling signaling.
Okay, cool. Thank you.
Yeah, thanks for the question. And so this is one thing that our lab is really interested in, is understanding the properties of this signaling hub. What's in there and what is it doing to control and coordinate the signaling of T cells from the receptor up to the decision making of, is that cell going to activate or not? We're going to watch the movie. And so if we are able to make some, understand a bit more about how T cells are tuned, how they're able to identify self from non-self, and how these receptors actually trigger to communicate information from the outside of the cell to the inside of the cell, we can really improve upon a lot of the medicines and therapeutics and diagnostic tools that we've already developed really clinically, and just looking at what happens. And so we could design engineered receptors in these cancer targeting cells that effectively fight the cancer without maybe acting too strongly and causing too much of an immune response that can lead to inflammation and such. And so we can if we know how the receptor is triggered, then we can design these alternative methods of triggering T cells in a more targeted manner. We can also improve upon treatments for autoimmune diseases. Autoimmune diseases often arise when, and inflammatory responses, when the T cell is too sensitive to self molecules and starts attacking our own protein fragments as if they're foreign. And this leads to inflammatory responses and really can degrade the functioning of our systems. And so by understanding how T cells are tuned, we could maybe improve upon these treatments. And then lastly, maybe build diagnostic tools to evaluate the health of a person's immune system, the health of their T cells and their ability to sense and discriminate self and non-self. And so with that, I would like to thank you. Since this is a general audience, I do want to note that you're taxpayers, and therefore your funding, your taxes go toward these government institutions that pay for this research. And so this really would not be possible without you in the audience. And I'd like to thank my lab, Jay, my mentor, and the others in the lab that really make this research on T cells possible. So with that, I will take any questions.
Great, thank you so much, Kiera. I have maybe a naive question about how this system works. It kind of makes sense to me how things it already knows are self versus not self would trigger this like time, the like stopwatch. But is it thought that basically anything new might also trigger it, or anything new that's kind of a specific structure, or right when you just like see something new, how does it know whether to trigger or not?
Yeah, that's a great question. So one thing that I did not get into is the variety of the T cells that we have and the diversity of T cell receptors that we have. So each T cell receptor can really only sense one or a few peptides. And we have T cells. Each T cell has its own receptor, and almost every T cell has its own receptor. So there's this huge diversity of T cell receptors that are floating around waiting to pick up some foreign thing. And if one of our T cells responds to that foreign thing, then there could be this immune response to it. And, you know, part of how we don't have our T cells reacting constantly to our proteins is that there is a selection process that the T cells go through so that the receptors that they're showing don't react to the self, but there's no way of knowing whether any given person's T cell receptors will react to something new. And so we may be seeing this with COVID right now, in that some people are able to, their immune systems, they just have cells that are able to handle it. And some people just don't have the T cell receptors that easily recognize the COVID virus.
That's super interesting. Thank you. All right. Well, I think currently we don't have any questions in the chat, but maybe Kristin, Kiera can stick around for a couple minutes, and people are happy to filter through and ask any questions if they want. We'll stick around for a little bit.
And Kristin had a question earlier that Olga asked too that we could circle back to. Wow.
Kristin, if you are available over there.
Yes, sorry, I was nodding, but I'm not able to share my video anymore. Yes, I'm happy to hang out.
Not a problem. Okay, I think the original question was whether there have been any unexpected or unintended consequences of reintroducing wolves to the Yellowstone or Jackson area.
Yes, that was the one I was starting to answer when my phone decided to stop working. Well, a lot of the sort of unintended consequences, at least in this region, have to do with how the wolves are interacting with kind of people and human wildlife conflict. And so there are, things like there are ranchers who are accustomed to having their cows out free range, who are worried about wolves eating their cattle, people worried about wolves eating their dogs and that kind of thing. And there also seem to be some potential effects sort of throughout the ecosystem. These elk are several hundred pounds, and wolves take down an elk and they don't eat every little piece of it. And so they're sort of providing a lot of more food out on the landscape for a lot of other animals. So they seem to possibly be helping other sort of scavenger species to have even more food available.
That's pretty cool. Do any of wolves' prey eat other things that are prey of wolves, if that makes sense? Like, can the wolf be helping one type of their own prey by leaving carcasses of another type?
Sort of. Yeah, it all gets very complicated, and it because it's hard to specifically study a bunch of different animals. We don't always have a clear idea of how this works, but there are certainly some indications in some places of things like, wolves maybe will attack coyotes. So in some places, the coyote population is down when the wolves are there, and then that makes the foxes do very well, because then the coyotes aren't attacking the foxes. But then in other areas where wolves have a lot of food and maybe don't attack coyotes as much, then they're actually helping coyotes, and coyotes are just sort of following the wolves and cleaning up after them. So there are these really interesting kind of different interactions in different places, and it's it'll take a lot more work ecologically to tease out like when those things do and don't happen and what governs those kinds of interactions.
Cool. And then it looks like there's a new question for Kiera in the chat. John Rose is asking if you've tried adding certain antibodies, for example, to soluble co-stimulatory molecules to see how it affects duration of interaction.
Yeah, that's a great question. And in our system right now, because we're using this synthetic supported lipid bilayer, right now we're, in my experiments, I do not add any agonist to the co-stimulatory molecules. So in my experiments, I'm not introducing co-stimulation to the T cells. But others in my lab do have co-stimulatory molecules on the bilayer, and they're taking very similar measurements, and we're comparing our data to see how the cells respond differently with and without this co-stimulation.
All right, cool. Thank you. All right. Oh, and then Olga is also asking whether the stopwatch model for T cell recognition explains why linkers trigger the immune system.
That is definitely one thought, because the linkers that people are making, and also the receptors that people are introducing to T cells and these engineered T cell receptors, bind very, very strongly to their agonist, to the thing that, to the thing that they bind to. And so we know, and one of the reasons why they're used is that if the stopwatch model holds, then the types of linkers that we're using would have that property. But there are other complications in other models about how the T cell activates. And So we can't be sure. I guess one example is, we have this lock and key sort of picture in our mind about how proteins work, how enzymes work, and how proteins bind other proteins. But then to use a molecule that binds a completely different part of that protein and to get the same response is very unusual. And right now we don't know if the response using these linkers is the same as the natural PMHC TCR response. But we use it anyway because it helps people.
Gotta keep using what works.
All right, and we have another question for Kiera from Facebook Live. So the question is there a longevity of T cells in the body that you have to look at? How do you study how long the T cells remain effective for? So hopefully that question makes sense.
Oh, so sort of like how long are they circulating and how long do we have that set of T cells?
Yeah, I think the question seems to be related to is there a lifetime to the T cells in order to be studied? And then, how long are they effective for?
Yeah, I really have no idea how this works in people and animals. And I'm a chemist. I study the chemistry side of these interactions. I don't really know. But I can tell you about the T cells that I use and that I study. So I do get my T cells from mice. I do have to sacrifice the mice and take their lymph nodes and isolate their T cells. And I'm able to do experiments on those T cells grown outside of the cell or outside of a living organism for about a week-ish is the time window that I have for the cells that I study. In a person, I have no idea.
All right, great, thank you. Well, if there are no other questions, I think coming through, then really we'd like to say thank you to Kiera and Kristin. These have been two really cool talks, and it's been really, really interesting to learn about your work. So we will have a seminar in December this year. So I think that will be on December, again, the third Tuesday. So that should be December 15th. Which hopefully will be early enough that we're all sticking around home for the holidays, but it still shouldn't disrupt that flow anyway. So more information will be sent out about that later. And also, again, just a huge thank you to your speakers and a huge thank you for everyone who attended today. So thank you, Kelsey, for helping us put this on. And it's been a great month showing.
Thank you so much. And thank you, Kristin and Kiera, for such excellent presentations. We look forward to what Popping the Science Bubble has to bring next month and in the new year. Thank you so much. And thank you, everyone. Have a good evening.
Yeah, you as well.
Yeah, thanks.
Yes, thank you.
Hi, thanks.